Remi, it's nice to have you here.
Okay, thank you very much, Arnie. Thank you very much for the invitation to be here. It's always very nice to be at the Institute, and all the more since I've heard that today temperature in Paris is actually close to 110, so I'm very happy to be here. It's much cooler also. So I'm going to tell you about some works, I would say, at the interface between physics or statistical physics and biology or computational biology. So maybe the format of the lecture will be slightly different from some of the other lectures you have heard. I will put more emphasis on equations, on computation. And what I am interested in is how actually you can predict or reconstruct or say something, extract some information about structure, fitness from protein sequence data. And also, if you are really able to do that in a very nice and precise way, then you should be also able to design new proteins which have sequences very far away from natural sequences with specific functions. And this is what the talk will be about. So I will start first with some basic facts about proteins, which actually may be totally useless for most of you, because maybe I guess you know a lot of things about proteins, but maybe some of you haven't heard too much about proteins, so it will be a reminder. And then I will talk about the conceptual approaches, two different approaches which actually merge in some way, and you'll see that at the end of the lecture. I'll talk about data, what kind of data we are looking at and we are processing. And then I go more in details into the methods and the application, some of the results. And then I end up with some perspectives about design and trying to give some numbers about the size of the protein world. So you'll see that later on. Okay, so let me start with some very basic facts about proteins. Okay, sorry, I have to learn. Okay, so what is a protein? It's essentially a polymer. It's an heteropolymer of amino acids. So there are different amino acids in nature. There are typically 20 amino acids whose chemical structure is seen here. So what is interesting is the variability here in the side chain group here, which may take 20 different values, or let's say with different physical, chemical, and genetic properties. So all these side chains amino acids will differ by their charge, their size, and molecular weight, their polarity. Maybe they have some hydrophobic or hydrophilic properties, so they want to be in contact or not with water, with the solvent. Their codons, how they are encoded from a genetic point of view, and so on. So typical proteins include from tens, there are small domains, to hundreds of amino acids, close to 1,000, for instance. And once you know the primary sequence of a protein, then this protein acquires some structure in space. And there are different levels of structures. The first level of structure is called secondary structure. So locally, some motifs may emerge. So these motifs are generally some local folding of a sequence onto itself through some weak hydrogen bonds. So two kinds of motifs are found in usually. So this motif here is called an alpha helix, where you see the polymer doing some kind of helical structure onto itself with an helix turn, which is about 3.6 amino acids. And another well-known motifs are the beta sheets here, when you see parallel or anti-parallel strands here, which are joined by hydrogen bonds and which acquire some kind of locally planary structure. And these motifs are connected by some more flexible loops, and the protein is able to fold, maybe assisted by other chaperone proteins, or maybe by itself, into a well-defined three-dimensional structure. Obviously, what I'm saying here is just an approximation, because this structure is not a static object. It fluctuates over time, so you have time fluctuation. But this structure is very important because it gives or provides to a protein its functional properties, okay? So how it will interact with other molecules and so on. So I will give you some examples of that right now. And I will focus on three different proteins during the talk, but let me start with the smallest one. It's not really a protein, it's only a domain of protein, which means usually it's included in larger proteins. It gives larger proteins some specific function. And the specific function here, given by this WW domain, is that it's important in eukaryotic signaling proteins. So what these small domains of about 30 to 40 amino acids is able to do is actually it's able to bind some ligands here. So here you see the three-dimensional structure. You can recognize the beta 1, beta 2, and beta 3 strands. So the three strands which are very similar to the one I've shown here. Okay, And it's able to bind here small ligands in the beta-2, beta-3 groove here. And one binding, the important binding site here is the tyrosine, which is shown here. And you can see also the side chains of 2W, 2 tryptophans. So W is the letter for tryptophans. And these Ws, these tryptophans, are highly conserved in this W domain, and they gave the name of the domain, actually. They are found in most of the sequences coding for this domain. So the ligands that this domain is able to bind to are proline-rich linear motifs, and they are known to be divided into different groups according to their specificities, okay? So different specificities, which are defined here. So type 1, times 2, 3, and 4. And you can see here the kind of motifs that these ligands should have in order to bind to specific domains. So you can have a rich proline ligand here with some tyrosine at the end and any residue in between the tyrosine and the proline. And you can see different kinds here. I mean, they are more or less well characterized from a biochemical point of view, but there are different papers, and not all of them argue about the kind of specificities these domains are able to have. So by specificity, I mean that depending on the sequence of the domain for the same structure, you may bind to type 1, type 2, type 3, and type 4. I'll come back to that later on. So now another example of a much bigger proteins with more than 600 amino acids, which is HSP70, the chaperone protein, which has many functions. Essentially, it can transport other proteins for degradation and bring them to proteases. It can assist protein folding, so it's a chaperone protein. And it does that by changing conformation. So here you see two of the conformations, essential conformation this protein is able to acquire. And so in different colors, you see the main, let's say, functional parts of proteins. So in red here, you see this, what is called the substrate binding domain, SBD. This is where other protein will bind. This gray part here is called the lead. So this lead domain can actually fall onto the SBD and trap the protein, another protein, okay? And the NBD here is the nucleotide binding domain. It's a domain where the ATP can actually bind and be transformed into ADP, and that will create and that will trigger the change of conformation. Okay, so it's an important notion here. It's these allosteric changes, huge changes of conformation. So the conformation of the structure of a protein is not unique. It would depend on the ligand. It would depend on the presence of ATP or ADP, and so on. We'll come back to that later on. Okay, so what are the important questions in a nutshell? So given an amino acid sequence, okay, this is a sequence for the WW domain. You recognize the same fold as before. This sequence actually will fold onto itself and acquire some well-defined structure. So in principle, the sequence fully defines the 3D structure and the corresponding function of the function of a corresponding protein. That is the kind of here the specificity and the kind of ligands it can bind to. So of course, as you saw in the previous slide, the idea of the concept of structure may be very complex. It's alterated about binding. There is all these allostericity properties I've just shown you. The idea that proteins have a well-defined structure is not always true. There is a class of very interesting proteins which are called intrinsically disordered proteins, which actually are very floppy and very, they don't have this crystalline structure that we are used to think about. But they may acquire structure when they bind to something, okay? So it's a very dynamical idea of structure in this particular case. The functions may be also very complex. You may have very complicated proteins with multiple domains, and they all take part of a global function of proteins. There may be different binding sites. The function may depend on the chemical context. It may depend also where the protein is in the cell, and so on. And also proteins may actually interact with themselves in the sense that you can have dimeric conformations which change actually the role of a protein, and so on. So it's a very complicated story. Another aspect I want to insist on is that the mapping between the sequence and the structure is also a very complex one. So for instance, this has been tested a lot. If you do some mutation, for instance, you change here some amino acid into, I don't know, another one, alanine here, then you can see what happens to a protein. So whether the structure and the function are disrupted or not. So most of the mutations you can think of are actually neutral. They don't seem to affect anything to a protein. But in some cases, the effect of a mutation can be absolutely dramatic. So there may be very highly detrimental mutations that the structure will simply unravel. Or maybe you will change a lot the function. Okay? So that's something which should not happen in nature, or maybe it could be useful for some evolutionary reasons in some specific context. But the point is that mutation may largely affect all the properties of proteins, in particular stability, activity, specificity, and everything you can think of. So this is something which is very important for evolutionary purposes, okay, because it constrains a lot the evolutionary path and history of a protein. But it is also very important for applications. So yesterday we had a talk by A. Boyden, and he mentioned at the end of the talk how he used actually evolution in the lab in order to produce specific proteins he wanted to have, okay? And this is used routinely by the industry. So you can do, let's say, artificial design of proteins using directed evolution, for instance, some kind of SELEX-like experiments where you have a target and select proteins, do random mutation of sequences, and select proteins which bind to a specific target or have some specific properties with microfluidics in order to select proteins. You can do also ab initio design, where, you know, if you really understand this mapping between sequences and structure, then principle and function, then in principle you should be able to modify the sequence and engineer it in order to meet the desired function you are interested in. So I think from a physical point of view, this is a very interesting problem because proteins obviously are extremely important from a biological point of view, but they are also physical objects. I mean, E. coli, the brain, are not physical objects. Well, they are to some extent, but they are really biological objects, right? They are extremely complex. But the protein is just a molecule. It's in the realm of chemistry and physics, right? It's not a living organism. It's just a molecule. So in principle, physics should be able to say something about these objects. And in fact, physicists have been trying for a long time to apply first principle approaches to model protein folding. That is, in principle, if you know all the amino acids along the sequence and you know the interaction between these amino acids, then you can write a gigantic Hamiltonian or energy function, and then you put it on your computer, and then you just simulate and find a ground state or something, okay? Let's say sample the configuration. The success of this kind of first principle approach has been very limited so far. And in fact, it has been okay in order to model very short segments, very short proteins, but it's virtually impossible to obtain, to fold proteins like the one I've shown before, okay? Like this one, for instance, or, you know, reasonable sized proteins, it's extremely impossible. So it's, to some extent, it's a failure of physics, okay? So this is why it's interesting also for physicists, because it's a complex system, in the sense where classical approaches don't work so much. So well, so why and how can we actually do better than that? I'm trying to master these things, but I'm not sure I'm able. Okay. So the idea that actually emerged maybe 2 decades ago, or even earlier than that, was actually to say, okay, even if we are not able to model a protein using, you know, Hamiltonian approaches or these kind of things, maybe we can use data which are available, which are given to us by nature, in order to achieve some kind of effective model for the protein. And so we have a lot of evolutionary data. And here you have this, you know, classical drawing of a tree of life with all the different organisms emerging from some last common ancestor. And if I think about a protein like WW or even HSP70, which is actually a better example, HSP70, this chaperone protein, is essentially found, or let's say some homologous version of this protein are found in all living organisms to a first approximation. So that means that starting from this last common ancestor, this protein has evolved along the trees, along the branches of a tree, and can be found in all the organisms. And to a first approximation, I insist this is not true, but it's only an approximated way of thinking, all these proteins in all the organisms have more or less the same structure, and they essentially have the same function. This is obviously not true if you think about it, because this organism lives in different environments, they are subject to different pressures and so on. But the structure is highly conserved, much more than the sequence. And here is an example of this conservation of the structure, which is found from this paper here, where you see as a function of the, let's say, so what you can do is that when you have, I will show you the data, how they can be found on a website, for instance, in practice, but essentially when you look at the different sequences which are homologous and code for the same protein, they are in the same protein family, then you can measure the distances between these sequences and see how the structure, when they are being established, they are being determined, differ or not. And what you see is that This is the root mean square deviation. So this is essentially when you have two different structures, you can try to map them onto each other and find the best alignment of structures. And you can do the same thing for the sequences. So you measure the distance between structures and the distance between sequences. And if you do that, you see that at the beginning here, while the distance between sequences along the x-axis is changing a lot, the distance between structures is not. And if you go above some threshold of the distance, which is called kind of twilight zone, that is when the percentage of similarity between sequences, let's say, is typically lower than 20% of the amino acids. Okay, so that means that 20% of the amino acids between two sequences are common and 80% are different. Then the structure they correspond to may be very different. So there might be a lot of diversity in the sequence space and not so much in terms of structure. So we say that the structure is conserved. So if we have all these sequences, then we have essentially that nature has sampled the space of possible sequences associated to one particular structure, and we should be able to do something and model these invariances. Yes?
So if the sequences are, this is maybe more general, but like the sequences are so diverged, like what is to say there's an evolutionary relationship so much as reemergence of the same function?
Yeah, I mean, sometimes you find this, I mean, it's very hard to say whether they are actually homologous in the sense that it diverge from the same common ancestor. It might be also that you find the same structure for two different sequences, and we know that it's just a convergent evolution to the same structure. They have no common ancestry property, okay? So it's a complicated thing. But let's say as long as I am here, there are a lot of similarities between the sequences, and I may think that actually they are homologous sequences, right? So I don't want to wander here. I want to stay here, yeah, which is a safe zone. It's easier, okay? I'm not very brave here, okay? Yeah.
More vague and general question. Is there any notion of like how often, like how many of these kinds of structures there are within these criteria conservation across?
You mean what is the number of folds compared to the number? I mean the. Yeah, okay, so I'll talk about the number of families, how people do, but the number of folds is actually much smaller than the number of families. So, and this is one of the reasons for the convergent evolution I was just mentioning. So that means that there are a lot of folds which are, I mean, not so many folds which are possible, which seems, or possible in the sense that they give rise to interesting and functional proteins. I don't know. I mean, people have characterized those folds by, I think, essentially brute force. By I mean, looking at the structure of all the kind of structure they could actually study and determine. But you had a very vague answer to your question. Okay. Okay. So what are the approaches we can think of in order to make advantage of these sequences here, and what we would like to do with that? So I will mention two approaches, and you'll see that actually they are essentially the same one, but there is just two different points of view. So we have some data like that. So these are sequences, okay, coming from humans, from mice, from Drosophila, or whatever you can think of, or different insect or whatever. And we assume that all these sequences are code for, let's say, almost the same structure, and they almost have the same function. The proteins have almost the same function. That's our basic assumption. So essentially, we are in this sequence space. So if the length of the protein is L, then the dimension, I mean, the number of possible sequences is 20 to the L. So there is a first problem here that all the sequences don't have the same length. Okay, the length is variable from organism to organism, but let me forget about that. Suppose it's a common length, L. We have 20 to the L possible sequences, and we have a few hundreds or a few thousand, a few tens of thousands of sequences which are good. That means we know that they code for proteins with the good and required structure and the required function. So in this sequence space, which is certainly not one-dimensional, but I just draw it this way because it's easier on the slide, we have these dots here which are the good sequences. And there are many, many other ones. So you see it's just a few points in a huge space. So what we would like to do is actually to have a model. So there will be some wide axis here, and you can think about the dimension here as a score. And this model here will define some kind of, for each sequence, you will have a value here, and it will define some kind of function like that, complicated curve, above the sequence space. And what you would like is that the constraint you have is that the good function, the one which are in the databases here, should actually correspond to peak in these functions, because you know there are good ones. And you don't know about the other sequences here. Probably they correspond to low values, but maybe not. I mean, because they were never sampled by evolution or by nature, but maybe they are good ones. So for instance, here you see this is a peak which is high, but there is no sequence like that in the database. So the question is, can we actually find some kind of model? And I'm thinking about a probabilistic model here, which will actually give to any sequence A1 to AL or AN, the length of a sequence, some probability, some score. So this is a very ill-defined problem, right? Because we just have the yellow dot or the orange dots here, and we like to find this green curve. So we cannot do that unless we put a lot of constraints on the curve, right? But suppose we're able to do that. Then what can we do with this curve? Well, we could repeat, I mean, we could compute many curves, one for each protein family, each structure function. And then if I give you a new sequence, maybe an organism you have just studied and you are able to sequence it, then maybe you can actually do some kind of query and say, OK, is it a sequence corresponding to this particular structure function or to this one and so on? This is what people routinely do with databases. They use maybe BLAST. But you could, if you had these green curves for all the possible known functions and structures, then you could see whether it is in a peak for one of these green curve, and maybe it's a bottom point in all the other ones, and you say, OK, this is likely that this sequence actually corresponds to this particular family. OK? So I will talk about that a little bit on. So what you would like to do is assign membership of a query sequence to one family. Another thing you would like to do is to infer structural invariances. If all these sequences here code for more or less for the same structure, then there is an invariance here. And this green curve in some way should reflect this invariance, okay? Which is a common structure and function. So how can we get that from the curve? And then you maybe would like to do more than that. Take, for instance, one of the sequences here, a natural sequence, and you would like to be able maybe to predict the effect of some mutation. As I told you, some mutation may be very neutral, nothing will happen. But the other one will disrupt completely the sequence, the structure and the function. So maybe we'd like to have some knowledge. If we understand that, we could test actually the validity of this green curve around some point here locally. This is a very complicated task because, you know, the sequences which are here in the database, they are very far away from each other. They come from highly diverse organisms. You know, you can have bacteria, you can have mammals, you can have everything you think of, okay, for some families. So they are very far from each other, so you infer some kind of global landscape, and then you ask the question whether actually this global landscape is actually giving me some information which is very precise locally around some particular wild-type sequence. And you will see, I mean, that people are doing that now with experiments. I will show you some examples. And then the last goal is, OK, can I use my model, if I really trust it, to find new sequences which apparently seems to have high score and test them experimentally? Are they functional? Do they have the right structure? OK, so this is the first approach. We want to find the green curve. The second approach is a bit more abstract. So let me look again at the sequence space. So now so before I have a 1D line, now it's a more three-dimensional things, but again, it's a very low-dimensional way of representing this L-dimensional space of protein sequences, where L is the length of a sequence. I have some dots here that correspond to all the natural sequences which are known, which have been sequenced. And these dots may have different colors because, for instance, if you think about the WW domains, they have different specificities. Depending on the sequence, you bind to type 1, type 2, type 3, or type 4 ligands. Now, can we actually make sense of these dots here and try to represent this space of sequences into a more abstract space, which I will call a representation space or feature space? Okay? So this feature space has to be defined. So I'm just plotting something which is not defined yet. You will see later on how to practically obtain something like that, where maybe you are able to extract some directions which have actually some biological meaning. So maybe one of these directions here will be the specificity of the WW domain. So for instance, H1 is a real coordinate, and sequences on the positive side here will be type 2 specific. And on the negative side, they will be type 1. Maybe this axis here will be the activity, whether actually the binding is actually efficient or not. So you may think that maybe you would like to understand how these sequences may be mapped onto a feature space. And actually, what we are talking about here is nothing but the genotype to phenotype mapping. Okay? So genotype being a sequence and the phenotype being the set of possible structural and functional features of a protein. Can we find this space and can we understand the mapping? So we'd like to have a mapping going from the sequence space to this feature space. And this mapping may be actually probabilistic. It would tell us, given a sequence here, to which region I should associate this sequence, to which region in the feature space I will be. Okay? And of course, this mapping should go both ways. That is, if now I am interested in a particular phenotype, for instance, a given value of the activity, a given set of specificity, and so on, then I would like to be able to understand where are the sequences in the sequence space that correspond to this particular phenotype. Okay? And in particular, for the design problem, what I would like to be able to do is maybe engineer some proteins with some desired properties, maybe a much lower activity or much higher activity. And this is what people do in directed evolution in the talk we saw yesterday. And then that should tell me where I should look at in the sequence space. Okay, so it's a different way of thinking about that. But you can think that, you can see that in some way, if I can forget about the feature space, that is, if I integrate out, and I'm just saying that in a very vague way, but I will be more precise in the following slides. If I integrate all of this, then this mapping should become some kind of effective representation or effective description of what is going in the sequence space. And that looks like very much the green curve I had here. So these are two different ways of thinking about the same sequence space. So what are the data? So the data are essentially sequence data we are interested in. So here is the number of sequences which are available on UniProt, which is one of the biggest databases in proteins, for proteins. As the year, here you see the number of sequences which are available. So now there are about 10 to the 8 sequences available. You saw a big drop. You can see a big drop here a few years ago. That the database had actually a lot of garbage, and it was cleaned. And people removed a lot of sequences which were actually double or not very, or corrupted and so on. But again, what is interesting here is that essentially the growth is exponential. So the number, or it's even maybe more than exponential. But essentially, let's say it's exponential. It's sufficient for comparison with the next slide. So the number of sequences is getting very large with time. The length of these sequences, as I said, range from 10 to, let's say, 1,000 amino acids. And they come from all possible organisms. So bacteria, eukaryotes, viruses, and so on. In different.
They're probably related. Many of these sequences, they're probably related. Let's say from different species, and very similar to each other. So
yes, absolutely.
Exploring probably. I mean, the number of effective.
Yes, absolutely. Yeah, so I will show. I mean, Pfam, which is a more organized and structured database. But I mean, I just wanted to show the total number of sequences, just brute force. There are other databases which are structural databases, and PDB is one of them. And PDB here I show exactly the same plot as before, but now you have you have the years here, and you have a number of structures. But if you can look at the numbers, they are much smaller. Obviously, there are much less structures which are known. Most of them known by X-ray studies and crystallographic studies. And essentially, if you look, this is the blue curve. Here, this is the number of new structures every year. The blue curve, and you see the growth is more or less linear. Maybe slightly quadratic, but not exponential, right? So the number of structures which are known now is about 10 to the 5 compared to the 10 to the 8 sequences. So it seems, if we just look at the numbers, it's a good idea to look at sequences in order to get information about the structure. And you will see, I will talk maybe just at the end of the lecture about RNA, where the situation is even more drastic. So many more sequences than structures. So there are other databases which are useful, and the database we use most of the time is Pfam. So Pfam is a database which is more organized than UniProt, at least that's my feeling. And in Pfam, sequences are actually clustered into families. These are the families I was talking about. So this is, for instance, what you get if you go onto Pfam, and this is just a screen capture. I mean, for the www domain, which is Pfam 00397, and when you get into this web page, you see the structure, you see the number of sequences which are available. So there are about 25,000 sequences available. I did that a couple of days ago, so I don't think that's changed a lot. Actually, this comes from September 2018. This is the last release. You see also the structures which are known, the interaction with other proteins, and so on. So how this is done, I will show you a very simple way of actually, when you have this huge database, of clustering them into families, because they are all related to each other, as you said. And if you go to another one, for instance, this is beta-lactamase. So beta-lactamase is one of these 18,000 families which are available on Pfam. This is a bacterial protein, which is very important because it confers antibiotic resistance to bacteria, for instance, to E. coli, to antibiotics from the family of penicillins or cephalosporins and so on. I'll talk a little bit about that later on. So you see, for each kind of protein family you are interested in, then you can get access to the information. And the information is this kind of thing. So these are sequences for beta-lactamases for different organisms. And so what the kind of format of the data you get are all the sequences. So each line is an organism, and each column is a site in the format. And the format is such that all the sequences of different lengths are aligned. And in order to do that, you have to you have to stretch the shorter sequences to make them the same length as the longest one. And this is done by introducing some gaps here. So there is this is not easy to align sequences. I'll just talk a little bit about that. But let's assume for a while that this is done, and in a nice way, which is not always the case, because it's complicated. The idea is that you had this last evolutionary ancestor, or let's say a common ancestor, and you may have actually deletion or insertion of some parts, and that would correspond to the different, that would define the different lengths of proteins which actually emerge at the tips of a tree. So if you look at this data, you see that some position here, some sites are highly conserved. So for instance, you see alanine on this site in most of the sequences. You see, for instance, proline in the other sequences, and so on. Usually, these sites correspond to the protein core. So inside the protein, this is a region of protein which is highly conserved. They may also correspond to functional sites. And if you change them, then it will change the specificity and the function of proteins. This is why it's highly conserved. And other sites, if you look, for instance, at the last at the columns here, seems to be highly variable. So they are apparently neutral. So this is the first approximation. So how can we model that? And this is where I'm going to the green curve again. So from the data, how can I get an approximation to this green curve? So the simplest thing you can think of, so that I would call it the simplest non-trivial model for protein sequences, I will assume that all these sites of proteins are actually evolving in an independent way. Okay? So it's a very drastic assumption, but it's an assumption which makes actually our life very simple. So if this is true, then the probability, so if I look at the, if I look at the data here, for each site I along the protein sequence, I can compute the statistics of the amino acids. So on each site I will have 20 numbers, 21 because there is a gap symbol, which tell me what is the frequency of this symbol on this site, which I call Fi of A. I is the site, A is the symbol, the amino acid. So if all the sites are independent, then I would expect that the probability of seeing a sequence A, which is A1 up to AN, is simply the product of the frequencies of the sites, right? So usually in physics and in bioinformatics, this is not written this way. This is written as exponential of the sum of I of GI-AI. So the GI is just the log of Fi, okay? And it's called a local field or local potential in physics. It's called a position weight matrix in bioinformatics. It's the same object. Okay. So for people accustomed with the statistical physics, you know about the Ising model. I think you all know about the Ising model. You know also about the Potts model. Yes, the Potts model is just a generalization to more than two symbols. And this is exactly what you would get for a local field, without any couplings between the sites here for a Potts model. Okay, so the log probability is just the argument of the exponential here, just the sum of the size of the GI of AI. And that I can rewrite in a slightly different way. I will introduce what people call in machine learning one-hot encoding. So I don't know if you are familiar with that. So one-hot encoding is that if you have A, so A is a variable which goes from 1 to 21. 20 amino acids plus the gap. So instead of having A going from 1 to 21, which is a little bit, difficult to handle, you introduce a vector here of dimension 21, which has all zeros except one entry, which is equal to 1, and that corresponds to the ace position here. Is it clear? So that means that I will use, it's a very, it's not a very ecological way of representing things, right? You use a big high dimensional thing to code for numbers. So that means, for instance, if you have A, which is, let's say, 1, that's the first category, so maybe it would be alanine, for instance, whatever. I mean, it depends how you order them. So then that you would associate the vector 1, 0, and 0, and there are 20 zeros here. Okay? And for maybe the gap symbol, which is the last one, then that will be the vector with all zeros here. The first 20 entries are 0, and the last entry is 1. Okay? So you can do that. So if you do that, you see just a way of rewriting the sum. And we'll define for each psi I, this one hot encoding here, you see now the sum over all the position, it can be written as the sum of the position on entries of these vectors. And that you recognize is simply a dot product between two vectors. One vector which I call G, which is a vector corresponding to, which defines actually this green curve. This is my model. And S, this is a sequence. And there are vectors in dimension 21 times the length of a sequence. So which means that my problem is a purely geometrical problem now. If I look at all the possible vectors, G1, G2, G3, G up to 10 to the 4, which correspond to all the vectors coding for all the protein families, they define specific vectors in this high dimensional space. What is the dimension of a space? It's essentially 21 times the length of a sequence. They are not all the same length, but the dimension is typically over the 10 to the 4. So we have 10 to the 4 vectors in dimension 10 to the 4, each one coding for a protein family. And now I have all the sequences in the database, and you would expect that these sequences, which are vectors, here I just represent the tip of the arrow here, there are dots in this space, and they would cluster along this direction. So they would have nice, large dot product along one direction, and very low ones along all the other directions. OK? Yes? Sure.
The local field is a representation of the interaction of the amino acid physically, right?
Well, I mean, here there is no interaction between the sites. It's just a propensity of having a given amino acid on one particular site.
Okay, so for the gap that you consider the 21
Yeah.
amino acid, is the local field is 0?
No. Well, I mean, it would be, this it would be on this particular site here. You have a large number of gaps. That means that for maybe half of the sequences have to be stretched here. So it might be a highly region of a protein. Typically, you find that when you have a protein which may be actually quite rigid, and you have loops which come out from the structure, and these loops may be short, they may be long. Okay, so you see a large variability. You will see in the following example that the length of a loop may be actually extremely important for function. Okay, it's not something which is random and nobody cares. It's important. You see what I mean about the length of a loop? So you got that by the presence or not of gaps. So now if I give you a new sequence, which is this green dot here, then you can ask to which family does it belong. And you see you just have to compute all the dot products with all the directions, G1, G2, G3, and so on. And hopefully one of them will be bigger than all the other ones. And you say, oh, that belongs to family 1. And this is exactly how Pfam works. I'm just cheating a little bit because I didn't say anything about how you align sequences of different lengths. It's a non-trivial task, which I've not done here. And in order to do that, you need something a bit more sophisticated, which is called a hidden Markov model, which is essentially trying to insert gaps and then until you reach a new site, which is one of these sites of this model, and then you kind of meet the symbols corresponding to the amino acids with these probabilities. I don't want to enter into the details. There are very nice books, in particular by Shanedi, explaining this very nicely.
If there is a completely new project that is coming from per galaxy and everything, it can tell you also that.
Yes, yeah, it will tell you. Okay, then it will tell you that all the scores will be low. So maybe you have to go into Pfam, and did that because I'm not one of the managers of Pfam, but you have to create a new page, and this is a new family, I guess. Yes, so there are unassigned protein sequences, absolutely. I don't know exactly what are the roots. I don't think that people do that if there is only one sequence. Maybe they have to find several of them. So, but exactly. Of course, you see that what I'm saying here doesn't always work, because if you go, maybe if your green dots is somewhere here, then it's bad. So maybe it could be that it's a new family you have to invent or to create. It may be also that it's a failure of the model. The model is very simple. It's a stupid model, right? It just counts a score on the site. And maybe, it's nice when you have highly conserved sites and so on, but as soon as it's become very far away and we get into this twilight zone I was mentioning before, then it's a mess. We don't know. Okay. So one thing also I just want to mention for those of you actually who know a little bit, maybe you heard about support vector machine or perceptron. Okay. This classification algorithm is simply a perceptron or support vector machine, essentially. Basically compute the dot product with some vectors and try to find the largest one. Yes?
I don't understand the fact that you are kind of assuming that this phenotype function is a continuous function in this sequence space. How justified is that?
This continuous, sorry?
As in, if it's not a continuous function.
Ah, you mean the green curve?
Yes.
Oh, okay. You're saying why should it be a continuous thing?
Yes.
Okay, so it's okay. So mathematically speaking, it's not continuous since my space here is discrete. I have only a discrete set of values, but I agree that the answer is not very nice. So yeah, so the scores here actually, so there is a discrete set can actually more or less have some kind of more or less fill in some kind of interval here in a close to continuous way. Let's say almost dense way. So what happens is that you will see actually, this is generally not true. I mean, in the sense that some sequences would have score which are essentially 0. In the sense that you cannot do this mutation, for instance, on this side. So it's a very bad sequence, should be 0. The model we use will never predict 0. You will see that concretely in a while. But it will predict a very low value, which is bad. So essentially what we'd like to do is to be able to fit the peaks here. And if the tails are not well fitted, it doesn't matter so much. They are so bad anyway. Why you want to predict this number with high accuracy? It doesn't even make sense, right? It's now not functional. So what we'd like to be able is to capture these peaks here. And okay, this is basically what the model is trying to do.
Yes, what happens if the inner product of one of these aspects is, let's say, equidistant to several keys?
Yeah, so in that case, you cannot say anything.
And that happens, or is something?
Okay, that happens if all these dot products become small. Okay? It never happens that you get two large products because these directions are far away. But when you get close to the origin, okay, then the dot products you get along this direction, for instance, the dot products become small and you don't know what to decide.
I can imagine with 10 to the 4, the chance of getting this probability of getting these events is high. Right?
Yes, you will see that actually we can compute that exactly in an abstract model of proteins. You will see that in a while. Yes.
It's probably very sensitive to working with sampling of the data that you start with.
Yes, you're right. You're right. So in fact, when you compute the statistics here, so for instance, this statistic fi of a, then here I didn't say anything about how that was computed. So the uniform way would be to give it the same way to all sequences. But what people do in practice, they don't do that. They try to do some smoothing. And when you have two sequences which are very close to each other, maybe you will give weight 1/2, 1/2. So you don't want to do overcounting, double counting, this kind of. So try to smooth a little bit. But you are right. I mean, there are a lot of biases in the databases. A lot of labs work on the same organism, so very close organism. Okay, so in fact, if we look more carefully at these sites where a lot of mutation happens, they are not neutral at all, because though they may change a lot from sequence to sequence, the changes between different columns may not be uncorrelated. There might be a lot of correlation between these changes. And this idea appeared at least 20 years ago, is that if we look at sites along the primary sequence which have there might be large correlation here, this large correlation could actually come from the fact that on the three-dimensional structure, these two sides may be close to each other. And if they are in contact, then you would expect that a mutation here would actually be compensated by some mutation here, but the mutation wouldn't be independent. For For instance, if you have a contact which is maintained by a plus and minus charges of the amino acid here, if you change a charge here, then you change also the other one. Otherwise, that would disrupt the structure, right? So the idea was put forward in the 90s that large correlation, pairwise correlation, could actually code for structural constraints. Okay. And in fact, you could have also other reasons for large correlation. For instance, if you have a ligand which is binding here on this part of the protein, then the correlation which, that would create some correlation between the values of the amino acid here, the identities of the amino acid, that because you have to bind in a specific way. You don't want to bind this kind of ligand, for instance. So there might be also functional sources of correlations. Okay, so what do people do in what did they do in the 90s? So what they did was simply to look, as I said, you can compute the first order moments, that is the frequencies of amino acids on the different sites, but you can go to higher moments and you can compute the joint frequency of having amino acid A on site I and B on site J. Okay, you can have access to that from the sequence data. Of course, there is a statistical problem here. I will talk a little bit about that a little bit later on. You have 10 to 100 amino acids in the sequences, so the size of this matrix is absolutely huge. Okay, so you have to extract a huge number of elements from this data. So the question is what is the accuracy on these numbers? Talk about that later on. So once you have this correlation matrix, okay, then if you had perfectly independent sites and you had very, very good samplings, all the elements, the non-diagonal elements, essentially or non-block diagonal elements should be 0. There will be no correlation. But of course, when you do that in practice, you don't see zeros. You see a lot of correlation. So what you can do is look, for instance, at the big elements in this matrix. Okay. So for instance, this matrix is a matrix where you have actually a dependence on the sites and so on the amino acids. If you want to have an indicator estimator which depends only on the pair of sites, you can actually project on all amino acids. So for each pair ij, I will compute the sum of the Cij AB squared, sum over AB, some kind of local Frobenius norm of this matrix. Okay. So now it depends only on the pair of ij, and the idea, and that was done in a paper 25 years ago, as soon as people had some sequences actually to work on. And if you do that and you say, okay, let me take all these indicators which depends on the pairs, let me rank them from the top one to the bottom one, and let me take just a small number of them, maybe the top 30 scores here, and see where they are, to which pair they correspond to on this protein. So this is a protein which is a Kunitz domain trypsin inhibitor. It's a small domain with 53 residues. You see that most of the top scores here actually correspond to pairs of amino acids which are far away on the domain. It doesn't work so well. So you get highly correlated amino acids. So the red ones are good in the sense that I think the criterion here was that the distance between the alpha carbons atoms was smaller than 8 angstrom, which is not a very stringent criterion actually. But you see, you get a lot of green ones which correspond to far away sites. So why do we get high correlation and no that correspond to residues which are not in contact? So that shouldn't be a surprise to people working in physics. There is a large distinction, a big distinction between correlation and interactions, right? We know you can have high correlation without any interaction, direct interaction. So what you can think of is that we should not look at the correlation matrix. We should work more and try to find some interaction network with some effective interactions. Whether they are not correlated to physical-chemical interaction is something else. Okay, we can talk about that, but at least they could be statistical interaction. And this statistical interaction should be inferred or calculated in a way that they reproduce the data, in particular the correlation matrix. So it's an inverse problem here, right? So imagine a network. The number of sites is the size of a protein, and you would like to find this interaction network. It's an interaction network where between any two sites, I and J, you can have 21 amino acids or symbols here, 21 here. So the matrix of interaction is a 21 squared matrix. And you would like to get all these numbers from the data. So compared to the model I had before, which was this model where you had a sum over all the sites, now I will have also a sum over the pairs of sites. Okay? So how this is done? So how this is defined first? So the model people consider, which is the simplest one you can think of in terms of statistical physics, is simply that it's a Potts model. but it's an unusual Potts model because usually in physics we have invariance by translation, for instance, these kind of things. Here we don't. Here we have just a bunch of sites with an unknown interaction network and unknown local fields. And that should define some distribution here. And so this is our model. So it's just a generalization to the non-independent site model of what I've done before. And so the coefficient Jij and Gi are unknown. And what I would like to do is to find them from the data. So how do we do that? We do maximum likelihood inference. So we say, OK, I have a bunch of sequences. So I will assume that they are all independent from each other, which is not true because they all come from the same phylogeny. But let's forget about that for a while. And then I would like to maximize this where the product is over all the sequences in the database. So it's a function of the fields and the couplings, and you would like to maximize this. Okay? So I'll talk in a while how this is done. But let me show you the outcome for the Kunitz domain. Okay? So this is what I've shown before for the correlations. And if you do the same thing, you compute the score from the statistical interaction, not from the correlation, you get a much, much better outcome. Okay? And this paper by Martin White and collaborators will actually change the field because then it was the idea that people had in the 90s was almost a good one. They were right to look at correlation, but they had to do a little bit of processing before actually exploiting the correlations. Yes?
Would you just give us a couple of slides? I was trying to write down yes, that, and managed to get it. Thank you.
OK. Yeah?
So the function that you're trying to maximize seems to be, like in physics language, the partition function and not the free energy per se. I'm confused about that when you're comparing it to Potts model.
OK, so if you look at this, OK, so look at what it means here. Maybe I should write it on the blackboard. So maybe it's better if I write the log of this thing. So what I want to maximize, so it says log P, OK?
Yes.
So it's a sum over all sequences. And then you see you get the sum over D. And so you get the sum over all pairs, pair interaction term, Jij. So this is AI, D. So this is the highest amino acid in the D sequence, AJ. You get a similar terms for the fields. OK? And then you get minus D times the log of a partition function, which is a function of all the G's and all the J's. And you want to maximize. So the J's are here and here. OK, so there have been. So I will tell you in a while how this is done in practice, but just to show you some application first. So actually, as I told you, this paper and all this work were very important because actually they gave rise to the best approaches for structural prediction from sequence data. So this is not solving the protein folding problem that Arnie was mentioning in the beginning because we cannot fold one sequence, OK? But if you have a lot of sequences, then you can say a lot about the common structure. So it's worse than solving the problem, but still it's very important, right? So it won the CASP11 competition where actually some people work on predicting structures before actually the experimental structures are published. So the CASP11 was not the last competition. There was the CASP13. So I say a word about who won for the CASP13, but CASP11 was 3 years ago. And it could predict with very nice precision, very good precision, a lot of structures which were not known at that time. So, and this is interesting because it's not only a structural prediction of some kind of static structure, but it's also structural prediction about how domains or let's say flexible part of a protein can interact together. And this is an example of a sensor histidine kinase where some mobile actually part of a protein can actually bind under the presence of ATP onto this histidine here, which is the activation site. And it was not known when the study was done. So the structure was known in this open configuration, but not in the closed configuration. And just by looking at the correlation between the amino acid in the sequences and doing this inverse problem, then people were able to understand where the statistical interaction and to actually have a predictable, to predict a model of interaction. It turned out that the structure was done later on, and it was confirmed by the structure. So it's a very interesting thing. A lot of things have been done about prediction of partners between proteins. So there are a lot of applications. Yes?
So are those on the right there, you have two domains of one, like...
Yes, one protein. Yes, absolutely.
And those sequences were always sort of kept together?
Yes, because it's a single protein.
One domain for a different sequence, can you also model the interaction between...
So this was done between different proteins, for instance, when you have partners and you would like to understand which partner is which part is interesting. What is the partner of this particular sequence? So there have been works on that here. I don't have slides, but I can give you some references also.
So it can be sort of made quite generic.
Yes, yes. But here, they were part of the same sequence, so you don't have to solve this association problem. It was easier. So there have been other applications, and I want to mention that on fitnesses. So I will be brief, but just to give you a flavor of what people are doing now. So there have been a lot of work on the structural prediction now. This is something which is more ongoing now. So I told you a little bit about beta-lactamase, which is a protein that confers antibiotic resistance to bacteria. And so beta-lactamase, it's very important to understand what is the fitness landscape of this protein. So what kind of mutation can actually kill this protein or not, or let's say diminish its activity. So people have done a lot of deep DMS, which is deep mutational scanning. That is essentially what you do. You start from the wild type sequence, and you try all the, let's say, 1-point mutation, the 2-point mutation, everything you can do in the lab. So here, this is an experiment which was done by a Tanayon group in Paris, when they try for all the position in a protein to do mutation, single-point mutation. And on each site, so you have the sites here, and you have different colors which correspond to the different amino acids. And this color corresponds to the change in fitness that you measure when you do the corresponding mutation. So how is the fitness measured? What people do, they just remove the wild type protein, they insert a mutated one, and they look at the growth rate in presence of antibiotics. And they use the MIC, which is the minimum inhibitory concentration, as a proxy for the fitness. Okay? The higher it is, then the more fit is the protein for the bacteria. And you can see a lot of blue and red bars, which correspond to decrease or increase in the fitness. Okay? So one question is, can we actually predict this kind of data? And this is similar, if you remember, to this green curve I was mentioning, and then you could zoom in some regions, which was the wild type, and you would like to predict locally what is the landscape. It's exactly this question. Okay, so how can this be done? Since we have a score for sequence, this green curve is a score, this log of probability is the log likelihood for sequence, then we can compute the change in score with respect to a wild type when you do the corresponding mutation. The change in score can be positive and can be negative. And you can try to relate this change in score to the change in fitness that people measure in experiment. And this is what is done here. This is a change in fitness, and this is the change in score. And so there is a lot of deviation. You can see there is some kind of trend here with the Spearman coefficient, which is about 0.7, which shows that even in the model is not perfect, it captures something. Yes?
So how do you measure the fitness? What units or what time? So because you say that the fitness is if the organism grows or not. But the fact that you have
No, fitness, it's not whether it grows or not. It's at which concentration of antibiotics it stops growing. So it's a number.
so these numbers are...
Yes. This number corresponds to the log of a concentration in some... I don't remember exactly the unit, but they try different concentration. They didn't do the whole continuum set of concentration, but they try different concentrations.
About the other axis of this graph, there's a lower energy, sort of a better performance.
Yeah, so maybe I made a mistake in the score here, just to be sure. So this is if a log... Yeah, okay, no, this is not the energy, this is minus the energy. This is the log of probability. Okay? So if you have an increase in that, that means the score of a mutant is higher, that means the energy of a mutant is lower, okay, compared to a wild type. So that corresponds to a higher fitness. I think the signs are correct. So this is not perfect. It works better than the independent site model, but there is some signal. But there is also some conceptual problem. A protein may have many different functions and aspects, and we have a single score. Okay? So this is one of the reasons you don't get a perfect correlation also, is that, you know, if you change the experiment, you will change the x-axis, but I cannot change the y-axis. That's bad, right? Something is not going well here. So there is a conceptual problem. So I talk about that. OK? So let me say just a few words about how the computations are done in practice. So how do we infer the spots model, these models here? Oh, that's bad. Not sure I want to. OK, let me try to. I don't want to send anything to Microsoft. OK. This is alive again. OK, so how do we infer this spots model? How do we infer this interaction and this local field? So there is a wealth of techniques, including, so you see, what is the problem here? The problem is that nobody knows how to compute z, the partition function. It's just an intractable problem, right? In general, for any kind of Jij, then we don't know how to compute z in a time which is not exponential in the length of protein. OK, so there are just a few cases where, you know, maybe one-dimensional structure, or these kind of things where you know how to compute, or for specific cases. So you can either try to bypass z, the computation of z, or you try to approximate it. And basically, there are two classes of approximation of techniques. So I won't talk about the second one, which are based on mean field or cluster expansion, these kind of things, which are interesting. Just say a few words about the first line. So the first algorithm to do that is Boltzmann machine learning. So probably you have heard about Boltzmann machine learning. So how does that work? It's just massive Monte Carlo. So you start from random guesses for the Ji and the Jij Maybe you can start from the independent model. It's very easy. You compute the log of Fia, and the J's are 0, some educated guess. Then you compute, this is in the one hot encoding notation, you compute the frequencies and the joint frequencies corresponding to your model using Monte Carlo simulation. That you have to run that for a long time in order to get precise estimates. You compare to the counterparts of these quantities extracting from the data, so you know exactly what you should have. Sorry, this is not Fij, this is Fia. It's a mistake, OK? So you compare. If you are lucky, they match, and that's it. Your algorithm stops. If not, for instance, if what you compute with your measure, with your parameters, is smaller than what you have in the data, then that means that probably you should increase the field potential. You should increase your potential. If the correlation is too large compared to what you have in the data, you should decrease the coupling. OK? And you just update that until you reach some nice approximation, and you stop. So it's nice because it's very easy to program. Problem is it runs forever, but OK, but you get something at some point. So there is a huge issue about the time it takes, especially when you have, I mean, you may be close to some critical point and things like that, so it may be very large. It's known to converge, because actually this is just a gradient ascent of a log likelihood, which is a convex function. So there is a guarantee of convergence, but not about the time. So that's one algorithm. There is a much more clever algorithm, which is called pseudo-likelihood method, which comes from statistical inference, which was not so well known in statistical physics. So the idea is the following. The idea is don't try to do exactly what you should do. Try to do something simpler. OK? So what you should do is maximize the product of these quantities over all the data. And that is bad because you don't know how to compute z. So don't do that. Try to look at a simpler problem where you look at one site on the protein and see how it interacts with the other ones. OK? So now the problem is that you don't want to infer the whole graph, but only some kind of star-like graph, which is obviously much, much simpler. So you look at site I. So you have to do it for I equal to 1 up to L, all of them, one after the other. And look at the interaction incoming onto I from the different sites, J, for instance. So now it's a one-side problem, because here you have the data you can put on these sites, and you have to compute a local partition function, which is only a sum of the 21 values of these guys, which is a very easy problem. So that can be done. And there are nice facts about this. So this is how you get, for instance. It's fast. It's polynomial in the number of data and in the size of the problem. This is not. It's asymptotically consistent. I think when d goes to infinity, if you have an infinite number of data, then you are sure you will find back the correct value of the parameters you would have found if you were able to solve exactly the problem. It's nice to know, but we never have an infinite number of data in practice, or it's a kind of satisfaction, but limited satisfaction. But there are some nice results. If the graph you have to infer is sparse enough, okay, then, and I don't want to say exactly the condition, but you can find them in this paper, then what is very nice is that the number of data you need in order to find a coupling of strength j is obviously larger and larger as j is small, because it's hard to detect a small coupling. You need more accuracy, more data, that's clear. So it has to scale, it scales as 1 over j squared. But the nice thing, it scales only as log n. That means it's a very small number. You can treat huge problems. If you double the size of the problem, it just increases a little bit the number of data you need. So it's a very efficient method from this point of view. OK, so there are just 2 methods, but of course there are many other ones. So there are other problems, other issues, which are statistical issues. So how many parameters do we have to infer? So we need 20 times n parameters for the fields and 20 squared times n minus 1 over 2 for the couplings. So if you do calculation for, suppose you have 300 amino acids for length, that gives 2 times 10 to the 7 parameters. It's a huge number. Okay, 20 million parameters you have to infer. Now, suppose you have 10,000 sequences, which is a very reasonable number of sequences you may find on Pfam nowadays. And you compute the information you have, assuming that all the symbols are independent, which is obviously completely wrong, they are highly correlated. But at most, you would get less bit of information than the number of parameters. So if you are extremely optimistic, you get 1 bit of information per parameter. You can decide what is the sign of the parameter. That's really bad. So how do people do that? Well, they put prior information. That's the only way you have. Prior information, that is, you have to introduce some penalties. So the penalties are typically L2 or L1 penalties. So I don't know if you are familiar with that. It's just saying that instead of maximizing this, which is the log likelihood of data, you say, OK, I have to make some compromise between maximizing this and the fact that I may use very large parameters or very large number of parameters to do this maximization. So I will pay a price for that. So you add to this some terms which, for instance, will, OK, so you want to maximize. So that will decrease, for instance, if your couplings here are large. So you may use large couplings in order to maximize, but you will pay a price for that. OK? So this is the L2 regularization, because it depends on the L2 norm of the matrix. And it's nice because it just shrinks all the couplings. You can use also what people like is L1. So L1 is nice because L1 close to the origin L2. So if you have a small coupling Jij, Jij squared is almost nothing close to 0. So actually the penalty with the 2 here doesn't give you anything. It doesn't constrain the value of small couplings. This one has a finite slope close to the origin. It's a cusp. That means as soon as you are above or below 0, then it grows very quickly. It's a finite gradient. Okay? So that is a way to constrain the Jij to have 0 values. So it's usually used in machine learning. So there are different ways of trying to shrink the number of parameters.
How did that pass out? People are in the statistical physics literature. There's some work doing L2 or L1, adding to the potential, some term like that.
Yes, to do the inference.
No, I mean in general, Hot models. I mean in the physics literature.
So in general, in physics, we always look at the direct problem. You are given the J's, Ij, and the Gi, and you want to compute the observables. So these Gij are fixed for, you don't do anything with them. So the only field of physics where this is done, I think, in statistical physics, is people are interested in disordered systems. When they look at spin glasses, so they look at random distribution of couplings of fields, and they look at what are the typical properties. So it's a bit like that, except this is quenched, while here this is not quenched. You have to change that during the dynamics of you infer your model. OK, so I'm afraid I don't have much time about that. So I wanted to say a few things about some conceptual problems. And one way, another thing is why should the Jij, large Jij, be in correspondence to contact? You know, I did this. There was this thing here. I did this thing here. It's nice to say, oh yeah, if they are in contact, there should be a large coupling. Yeah, okay, but I mean, it's not a very strong justification, right? You would like to understand better that. In fact, the situation is quite is interesting because you could think, okay, you could think this is not obvious, right? Yeah, I wanted to say something about that. It's just a distraction here. So, you know, if you have a protein, there are many contacts. But you could think that maybe some contacts are extremely important to maintain the structure. Other ones are the consequences of the first contact. So if they are not very important, then there should be no covariation between the corresponding residues. So the Jij should be very small. So what are the important contacts? And there might be actually big Jij which are not related to the structure. So the connection between large Jij and structure is not obvious at all. So in order to understand that, we looked at lattice proteins, which are, it turns out that looking on the web, I've seen that there is some work from people at the University of Hull in the UK, which proposed some software on Minecraft in order to model proteins. I've seen that. But in fact, what we did was inspired from the 90s. So I just want to show that very quickly. They were inspired from the 90s from these kind of cubic proteins. So in the 90s, people were very much interested in biophysics about proteins and protein design. But of course, the power of computers was not the power of now. So they did very simple models on cubic lattices. It turns out that these models are quite interesting and exactly solvable. So you can do a lot and try to build a protein world and with this model and see what happens. So it's a very nice way of benchmarking these approaches. So if you are interested, I can tell you more about that, but I don't have time now. Just as a very brief things, I mean, all the kind of approaches you can have, I showed you on real proteins, can be applied to that, and they work well. And they tell you also that you can have big Jij. So in many cases, big Jij correspond to contacts on the native structure. But in some cases, and that corresponds to what people call positive design in proteins. So you want actually to favor energetically contacts on the native structure. But other Jij may actually correspond to what people call negative design. You have to understand that there are many protein structures which are in competition. A given sequence can fall in many different ways. Okay. So what you have to do is actually to be sure that it will fall in one specific way and not in the other way. Okay, in the other structures. And that you have to do some fine-tuning, which actually has nothing to do with the contact on the native structure, but just to avoid the other one. And you may find some big Jij which correspond to that. Okay, I'm just skipping all this stuff here. Okay, so now what about the other conceptual issues? I mean, it's nice to do this spots model because it looks like statistical physics, and we're happy with that. But it doesn't make so much sense because what are we doing? We are fitting all the pairwise correlations in the data, and probably most of them actually are not meaningful at all, not important. And we are not fitting any higher order correlations. But you may expect that triplets of amino acids or maybe quadruplets may actually be very important in some situations. So how can we do better than that? Another thing also I think which is known is that people in physics usually are given the coupling for free. When in physics, you are given the Hamiltonian. Here you see, I mean, the whole thing is about trying to get the Hamiltonian from the data. But in physics, you are given the Hamiltonian for free, and it's not over. Once you get the Hamiltonian, understanding what the system is doing is difficult, right? And here, even if you spend a lot of time to infer all these coefficients, you are still far away from understanding how the fitness landscape is done or made up. You don't know what is going on. So for instance, if I tell you all the pairwise interaction, how do you understand what is a collective excitation or a big piece of amino acid you should change in order to move to another conformation? It's very difficult. So knowing the graph doesn't tell you anything directly about the fitness landscape. It's very implicit. Okay, so another idea. So now I move to the approach number 2 that I mentioned at the beginning, which is trying to find some representation space. Okay, and this is an idea which comes from machine learning, which has been actually comes from neuroscience, and that related to the idea of representation we heard about yesterday afternoon. And here it's translated in a very, let's say, it's a kind of caricature of what we heard about yesterday. I mean, it's a very simple idea of representation. So the idea is that instead of looking at a network of interaction between sites, you may think that actually you have your different sites, and they actually are subject to some constraints, okay, coming from some hidden variables. And these hidden variables will actually code for the features of a protein. So these hidden variables can tell you whether you are in group 1, type 1, type 2, type 3, type 4, whether you have a contact between these two residues or not, whether this part of a protein should be in an alpha helix or it should be a beta strand or whatever, all the kind of constraints you can think of, right? And these variables actually produce some effects on the amino acid sequences and determine and shape the distribution of sequences. And you would like to find them from the data. So, one way to do that is to use what people call restricted Boltzmann machines. This is actually this is part also of what has been developed over the past years in machine learning under the name of autoencoders. Okay, it's very much related to that, but I think this is much closer to the POTS model, so I prefer to use that in this lecture. So remember what we had in the POTS model, we had terms, local terms on sites or interactions. Now I will unravel this interaction and introduce a new set of variables, which I'll call hidden variables, which are the h mu. So the number of hidden variables is not fixed now, it's just a number. And what we have now in the graph, we don't have any more interaction between the sites, but we have only interaction between the two layers. This is the layer, which you call visible layer, where the sequence is, and this is the layer of these hidden mysterious variables here. And they do interact through some couplings, which depends on the site I here on the sequence, the site mu in the hidden layer, and the amino acid AI, which is carried by the site I in the sequence. Okay? And you can see here some local terms on the site in the sequence, some local terms acting on these hidden variables, and the coupling between the visible and hidden variables. Is it okay? Yes?
Can you treat them as a bath for the rest of the system, like proteins?
In some way, yes. Yes. Actually, yeah. Actually, if I want, I may be interested in different things. This is define the joint distribution of the sequences and these hidden variables. I may actually integrate out formally over these hidden variables, and I get a score for the sequence only, which is what we need in order to maximize. We want to maximize this over the data set, right? But this approach, this formulation is more powerful because that defines also the conditional distribution of H given A. And that tells you, remember this mapping I had between the sequence space and the feature space. So P of H given A is actually doing this mapping. It tells you how, from given a sequence, where you should be in the feature space, this hidden representation. And P of A given H is what kind of sequences corresponding to a spot here in this representation space. And P of A is just the marginal distribution here. Okay? So what we have to do now is to train the machine in order to learn these fields here as before. So before we had to train the J, you had to get the potential on the interaction. Now we have to get the potentials here and here, and also the interactions. How this is done, this is just an extension of Boltzmann machine learning. This is exactly the same idea, except that Boltzmann machine learning was trying to match the correlations given by the model and the correlation given by the data. And here you have to match the correlation between the hidden and the visible variables in the data and the one given by the model. But it's moment matching algorithm. It's exactly the same. There is no pseudo-likelihood algorithm for RBM so far because it's difficult. So people do intensive, learning. But it's a simple machine. So it's not a deep machine, one hidden layer. So one thing I would like to say briefly is that this machine, if you choose that the potential U of H, this hidden potential here, is a quadratic potential, then you can actually compute, when you look at the marginal distribution, you can compute it. You can integrate over the H's because it's just a Gaussian distribution, and you get exactly what? You get exactly the POTS model. So this is a machine which includes the POTS model as a subcase. If U of H is not a quadratic potential, then it describes something more complex. In particular, if U of H is not a quadratic potential, it can code for higher order interaction between the visible units, which is something you really need to capture. So for instance, the black potential is the usual potential corresponding to a POTS model, and you can have the blue one, you can have the red ones, you can have different kind of potential, you can parameterize with a few parameters, and you can describe much power machines. So in the 5 minutes which are left, let me just show you some examples and of what we can do with that. So you can train, for instance, this machine with on the data of the W and the WW domain, the HSP70 chaperone proteins, and so on. And what you get, so remember what these machines, okay. What you get are this, I will show you only these weights here. Remember, what is interesting is how each hidden unit actually is seeing the visible units, what kind of feature it codes for.
How do you determine the dimensionality of the internal space there?
Okay, so we do that by cross-validation. So I will show you, I'm afraid, during the break, because I don't have to. Okay, essentially, so let me just show you the representation. So here is the site I, and I'm showing one graph for each mu. So this is the first hidden unit, the second hidden unit, and so on, and there are 100 of them, okay? So for each site I and each mu, you have the amino acid to choose, A. So there are 21 possibilities, and I'm showing that with the letter code. Given the letter corresponding to the amino acid, the W is just proportional to the size of the letter. It's just a way to see a matrix, you know. And it can be negative if you have a negative weight, or positive, it's above 0. So you see some weights which are actually 0 everywhere except on 2 sites. So this particular hidden unit is looking at only 2 sites on the sequence, you know, and it wants to be either to be activated very strongly positively or very strongly negatively. And the way you have to do that is to have either, for instance, if you look at the negative side, on this side 22, you have to add lysine or arginine, and here you have the aspartic or glutamic acid, okay? And these are amino acids with opposite charges, and it's a way to maintain a contact on the structure. So there are hidden units which just impose structural constraints on contact. They are very similar to the GIJ we had before, to a pairwise contact. But you may find also more subtle structural constraints, which are, for instance, correspond to the end and the beginning of these beta strands here, and there are codes for more than simple pairwise contact, but you know, 3 amino acids which are taken to sandwich 2 other amino acids here. So a more rigid constraint. Okay, so we have a catalog of different modes, features which can be obtained. This one is nice, that is at the end of the chaperone protein here, at the C terminal. And you see there is a long, so the weights are almost 0 everywhere. And so there is one hidden unit which is looking at the tail of a protein. It's making sure that either you have small tiny hydrophobic residues here, or you have a charged tail, either all pluses or all minus charges, essentially. And it's known that the tail of protein is very important to interact with other proteins. You can have a dimeric association, for instance, or interact with other factors which are important for the regulation of the chaperone. So you can see a lot of modes like that, corresponding, for instance, to allosteric changes and so on. So I don't have time to explain. I mean, if you're interested, I can explain to you in detail this mode. So maybe, and they are quite interesting, there are modes also which involve a lot of gaps. So you say, okay, why do we need to look at the gaps? So whether you see these gaps here, that means they control the size of the loop of these flexible parts of the protein. Okay? So if you activate positively this hidden unit, that means that the loop is long, and here the loop is very small, it's very short. And you can see that actually the sign of the activation is correlated to the function of the protein. Okay, and so that means clearly it's a way to see some features which are related to function. So I'm afraid I'm just getting over time. So I had a lot of things to say about why we think this machine works. So just to answer your question, and then I will show you some perspective. So how do we choose the number of hidden units, the strength of regularization, and so on? In principle, what you should do, you should you should take your data, take a part to do the training, another part to do the test, and you should maximize the log likelihood of the test. Maybe minus some regularization terms on well. If you do that, so this is, for instance, the training here, log likelihood, and the test log likelihood as a function of a penalty, for instance, but you could do the same thing as the number of hidden units. In principle, you should be here. If you look at what you have, for instance, a given hidden unit here, you get these kind of things here. It's very hard to interpret, and you don't see anything. The machine seems to have the best performance here, but you don't understand anything to what is going. But the thing is that there is a large plateau with very small loss in log likelihood, when you can increase a lot the sparsity. If you increase too much, then it drops, because the machine is not powerful enough, where you get much nicer features and much more interpretable features. And so we have some theoretical understanding of where you should go in order to do that. But I think it's important to do. We could use big machine learning technique, big black box, and just put it, and you get large performance, but it's very hard to understand what is going on. Here it's a small machine, but I think we can try to interpret what it's actually extracting from the data. So in the last maybe 2 minutes, I just want to say a few things about perspective. So things which may be interesting is really the real test of the power of this, I think, of these models is whether they are able to actually help you design new proteins. So let me forget about my Minecraft thing. So this is an idea which actually is not recent, and there was a beautiful paper by Rama Ranganathan about 15 years ago or 14 years ago, where he did exactly that for the WW domain. So what he did, he actually generated random sequences of a WW domain, maintaining the 1 point and the 2 point statistics. But there are sequences which are not natural sequences. Okay? So essentially, he didn't do it exactly the way I'm saying that, but it's more or less equivalent to sampling the POTS model inferred from the WW sequence data with the Monte Carlo algorithm. You generate some sequences, and you test them experimentally. And what he found is that if you keep only the 1 point moment, so that there is no interaction, you generate sequences which are very bad. They don't fold. If you keep also the 2 points, then a large fraction, which was about 30% of the sequences, were able to fold in the right fold, the natural one. I'm trying. So I think I will stop here. And here are the few conclusions, so I will just read them. I think the data-driven models for proteins are interesting. That's my non-subjective point of view. Because they may give access to structural, functional, and evolutionary information. I didn't say too much about that, but okay, I have some slides about that if you're interested. And they represent some kind, I think, ideal testing for control machine learning approaches. And also for trying to solve a non-trivial problem in physics. I see it as the boundary of physics and boundary of biology where they meet. I think there are a lot of biological aspects which are interesting. So better characterization of a space of feasible sequences. I'll just talk a little bit about that. Probably also, if we have this representation space, then we can think about transition paths and evolutionary paths, because then evolution is supposed to act on the, phenotype, and so we could see whether actually possible path in the phenotypic space. What does that mean in terms of constraints in the genotypic space, in the genotypes, for genotypes? And the last thing I would just mention is that some of you are interesting, is that in principle, RNA are other biomolecules, and they are not so different from proteins from a computer science point of view. You have a primary sequence. Instead of having 20 symbols, you have 4. It's even simpler. They are most of the time much shorter. We have databases of RNA sequences. We have also some structures. But so far, actually, this kind of approaches the success is limited on RNA. And there are many reasons for that. First of all, there are not so many structures which are available. Conservation in RNA is much higher than in proteins. And the secondary structure is essentially, the signal is so strong, it basically kills the tertiary structure signal. So there is a lot of space for improvement and trying to apply these techniques for RNA, and that would be extremely useful also for designing RNA for better understanding the structural properties and functional properties. Okay, so I will stop here.